All right. So I'd like to say a few introductory words. I'm Erik Winfree. I'm one of William's thesis advisors. The other is Richard Murray. So William comes to us, came to Caltech, originally from Brown University, where, among many other things, he studied polymer physics and the application to mass spec. And later he spent some time at the Institute for Systems Biology in Seattle, where he did some statistics with applications to cancer. And finally joined us at Caltech as a graduate student in the Computation and Neural Systems department. So one might ask, for someone whose final thesis is chemical reaction networks, majorly in the title, why CNS? Well, partly this can be explained that William's interest was not just in how to do inference about biological systems, but also came to encompass how biological systems perform inference. And he's going to expand on that theme quite a bit later today. Another sort of complementary balance that William found at Caltech was having two advisors, Richard Murray and myself. So he got to sort of split his time between a lab that does theory and experiments around real biological systems, and one that does theory and experiments around completely non-biological molecular systems. And he put that together and sort of used that as a fertile ground in which to weave his own story. And I should say, to some extent, he has a third advisor, Manoj Gopalkrishnan. So one thing that William did during his time was spend several months on an NSF fellowship in India, working in Mumbai with Manoj Gopalkrishnan. So you might get the impression already that William is a very independent scientist already, making his own decisions about what to pursue, and that has certainly been one of the main themes of his time here. He's been very passionate and very fierce in his pursuit of the things that he's really interested in, and that's has led to quite a compelling story, I think. I'm going to conclude my comments with an explanation of what you're looking at right now. So our lab has a tradition, like many labs, of people in the group making a poster for the graduation, the defense event. And at the moment, my group is fairly small, and the only person who really knew William very well was Andres Ortiz, who's already left Caltech and is in the Santa Fe Institute. But he happens to be an artist, so he had his own artistic vision here, knowing that William is fond of some Eastern philosophy. This is intended to be a Mandela of the spirit of Boltzmann machines. So you can see a little bit of nodes and networks and connections, and you'll see why that's appropriate for William's thesis as time progresses. So I will pause just for a second and ask whether Richard wants to say some words of introduction too, as the other advisor.
I will say nothing, except I'd love to hear what William's going to tell us. So let's turn it over to him.
All yours, William.
Thank you, Erik and Richard. Great. So as Erik already said, I'm going to be talking about compilation and inference with chemical reaction networks. Here's another piece of art by Andres, this one in the figure. And he has quite a way of sort of showing things, and I've co-opted this one to sort of give you a perspective on what you want to be thinking about during my presentation, which is how do cells and biochemical systems think? And in order to sort of begin or to begin giving a perspective on this, my PhD is drawn from 3 areas: statistical physics, going back to my undergraduate days, although in some new forms, machine learning, and synthetic and systems biology. And when you sort of put these together, one can maybe start imagining how biochemical computation works in a holistic way. And I'm going to give you in this presentation some thoughts, ideas, and results in that direction. And so a brief outline is I'm going to start with introduction and go over some of the key ideas in each of these areas. I'm then going to talk about software that I have created during my PhD, some theoretical work during my PhD, and finally some experimental work. And I'm going to conclude trying to tie all of this together. And if you have short clarifying questions throughout, please feel free to ask. If you have longer, more elaborate questions, I prefer you hold those to the end. And if you'll notice, there's slide numbers. And so you can just look at the slide number to refer back to it for a future question if you need. So I'm going to do something a little bit crazy and try to explain machine learning, where there are, thousands and thousands of papers in one slide. Let's take a go at it. So a machine learning algorithm learns by parameterizing a function with data. Now, what does that mean? I have some data, which I'm calling X, and I have some function F, and I'm going to choose parameters, W, that minimize an error. And so you can write this as an optimization problem. And the art of machine learning is not writing down the problem, but rather it's carrying out the learning and choosing a function, F, that really can do what you want it to do. And so a couple of examples for those of you who have background in machine learning of this framework is, a linear function is a choice of F, it's a line, and my algorithm might be least squares. If I'm doing something with a deep neural network, my function is now, this complex set of interconnected sigmoids, and my algorithm is backpropagation. If I'm using something like a Gaussian mixture model, maybe I use the expectation maximization algorithm to fit it. And as I mentioned, the art of machine learning is choosing F and also, figuring out how to do the number crunching to actually solve for W. I'm not really going to be talking about either of those things in this presentation. Instead, to me, machine learning is an inspiration for how biological systems and chemical systems might compute. So unlike traditional programming, where you write down a bunch of computer code, if statements, then statements, loops, logical conditions to get an answer, machine learning computes with a function F, and the coding is done by choosing numbers, parameters for that function. And you might imagine that a physical system like a cell, which is governed by the laws of physics, has some complicated equation that governs how it behaves, and there are parameters F, and we can interpret that as a type of computation. And I'll elaborate more on how machine learning is connected to chemical systems later. Now, the sort of inspiration behind all of this, I've said the word cell many times. I'm clearly thinking about biology. And in particular, I'm thinking about systems and synthetic biology. So what are these? Well, I think the inspiration the comparison is best by thinking about what's happened in the last 100 years. In the 20th century, we saw information become incredibly accessible with radio, then TV, and finally the internet. Basically, information became free. I think the 21st century has a promise for similar revolutions, but they're going to be through biology. And we're going to see breakthroughs in healthcare, nutrition, sustainable technologies, energy, which have the potential to democratize access and production of material goods. And the two fields that I think will allow this are systems and synthetic biology. Systems biology studies biology quantitatively as interconnected networks. So instead of thinking about one molecule or one organism, I'm going to look at networks of molecules and how they behave, or ecosystems or organisms. And I'm going to try to use numbers and mathematics to really quantify this complexity as a systems biologist. Synthetic biology, on the other hand, is an engineering discipline, which tries to repurpose biological systems to do new things. And really, I see these as two sides of the same coin. So on one hand, you can't engineer something if you don't know how it works underneath. So you need systems biology to effectively deploy synthetic biology. Synthetic biology also offers the ability to probe biological systems in more complicated, nuanced, and high throughput ways than we have ever had in the past, allowing us to understand increasingly complex biological systems. So you can sort of see a feedback loop here, and my hope is that in the next, say, 50 or 100 years, we're going to really see this exploding in something as amazing as the internet. Now, in order for this revolution to happen, I think systems and synthetic biology as fields can learn a lot from machine learning. And interestingly, machine learning was inspired in part by biology. So I'm going to show you a timeline of sort of how the machine learning revolution that's culminated in the last 20 years in some pretty cool things happened, and try to explain how that relates to the work I've done in biology. So if you look back in the 1950s, you have sort of some of the earliest machine learning prototypes, you might call them. The perceptron, I think, is particularly interesting. This is an early neural network that was built out of analog cables, and it's inspired by the brain. And these are sort of predecessors to today's deep neural networks. In the 80s, you had some sort of a resurgence of theoretical ideas. You're going to hear a lot about Boltzmann machines later. The backpropagation algorithm was rediscovered and found to apply to a version of the perceptron that we now call a deep neural net. And so you really see this theoretical birth of many of the ideas, not all, but many of the ideas that have made modern machine learning possible. However, it wasn't until the late 90s that there started being enough data to actually take this theory and test it, compare it, really understand how it performs on real problems. And the MNIST database, I think, catalyzed a lot of this. It's a database of just the numbers 0 through 9, images of them, but a lot of images, 60,000, and that really allowed all of this theory to be tested, compared, and understood in a more sort of empirical, pragmatic way. Of course, with big data and complex theory, you need software to do the number crunching. So in the early 2000s and mid 2000s, you have the first set of sort of specialized, easily accessible machine learning software. And then more recently, we've had sort of another round of software development where people started using GPUs, graphics processing units, that have really made machine learning much, much more efficient and scalable. Now, finally, you see some sort of success stories coming out of this. IBM Watson was an early success story in machine learning, winning Jeopardy and chess a few years before. And more recently, I think the people at DeepMind are doing unbelievable things. You have AlphaGo beating the Go game, which is much harder than chess. And to me, as something of a biologist, AlphaFold is amazing. This is software that predicts protein structure from sequences. It uses machine learning and deep neural networks to do this. And if you had asked me 5 years ago, would protein structure be predicted by a neural network in the next decade, I would have told you probably not. And I was wrong. AlphaFold has done a job that surpasses anything I would have expected. So the key here is that I want to bring the sort of same three things, theory, data, and software, and many people are trying to do this, not just me, but my PhD was focused around here, to systems and synthetic biology, and show how they can really be used and inform each other. So in my talk, I'm going to talk about software, theory, and experiments. But first, I have to sort of talk about the mathematical underpinnings of my thesis, and this is chemical reaction networks. So these are a mathematical model that are rooted in statistical physics and are used to represent systems in synthetic and systems biology. And I think it's really important that this is both a rigorous mathematical model and that it comes from physics, because we know that anything we can say about CRNs is going to be, fairly broadly applicable to many different biological systems and can be physically realistic. So formally, what is a CRN? It's a set of species. Here I'm calling them X. You'll see other names later in the PowerPoint. And reactions. And here I'm using some complex notation, but you can see that we have some inputs. These are the I's, the input species, go to some set of outputs at some rate rho sub r. And I'll give you some examples. So CRNs are very expressive. Maybe in high school chemistry, some of you saw this equation where oxygen combines with hydrogen gas to form water. This is how hydrogen burns. This is a CRN, where the rate constant is sort of the rate of burning. You can also see CRNs in molecular biology. If we think about a gene that produces a transcript, and the transcript in turn produces a protein, this is transcription and translation, you can encapsulate the central dogma of biology in a two-reaction network, although this is very simplified. And you can think of CRNs even more abstractly. So here we talked about sort of atoms and small molecules. Here we're talking about large molecules. You can also use CRNs to talk about more abstract things like bunnies. So if a black bunny and a white bunny come together and produce 5 new gray bunnies, I can write that as a CRN. And the fact that CRNs can express so many different types of things makes them very versatile. You can use them to model lots of elements and facets of what's going on in biology. Now, I've sort of shown you these chemical reactions, but these are also have a rigorous mathematical interpretation. So they're a way of writing down equations, so to speak. And they have 2 types of dynamics. The first is deterministic dynamics. And so here we're talking about the concentrations of chemical species. And concentrations are real numbers, and you get a set of differential equations given by this formula, which can convert any of those reactions I showed you into ordinary differential equations. So here's an example. We have A goes to 2B, and what you see here is that A starts at some value and goes down, and then B increases. You can see this in the dotted line, sorry, dashed line. You can also talk about stochastic CRN dynamics. And in this case, if you imagine zooming in on a very, very small volume of molecules, you'll notice that, well, molecules don't come in halves. They come in discrete entities. There's 1 molecule, or there's 10 molecules, or there's Avogadro's number of molecules, but they're not coming in 1.37 molecules. So in stochastic CRN dynamics, you model the counts. X is a positive integer of each species, and you get a set of differential equations for the probability of a count. So you can see some example trajectories here. These are the solid lines that sort of are grayed out, are showing example stochastic trajectories for the same chemical reaction network. You can take any chemical reaction network and interpret it either stochastically or deterministically. And when you do the stochastic treatment, if you average over all of these trajectories, you actually get a probability distribution, the probability of having some number of counts that changes in time. And I want to highlight over here this distribution on the right. You'll notice that has this striped behavior. This is because B always comes in units of 2. So if I start with 0B, I'm always going to have even numbers of B, and I'll never have an odd number of B. And this complexity, the fact that B cannot have any value, that B has sort of a restricted number of integer values, is something that's going to play a predominant role later. So just sort of putting that in your minds. But before we get to that sort of complicated theory, I want to talk about some software. And I think software will sort of expose why CRNs are such a useful model. And one of the many software projects I worked on at Caltech is called Biocompiler. And this was joint work with Ayush Pandey and Andre Shore and Zoltan Souza and Richard Murray. And so what is Biocompiler? This is a software package. Well, everything's in the name. So bio stands for biological. It's a library of biochemical components and mechanisms. The CRN, of course, stands for chemical reaction network. I just talked about those. Compiler is from computer science, the idea of taking a specification and generating something more complex. So I'll describe more about compilers in a second. And the Pi is for Python. Biocompiler was built to be integrated with other existing tools and pipelines. It's not meant to stand on its own. It's meant to be part of sort of the greater scientific programming software community. So what is chemical reaction network compilation? Well, let's start with compilation. Compilation converts a specification to an implementation. So an example of this is computer code. You say maybe write down some C++, and it compiles into machine code, maybe binaries, 1s and 0s. Despite what the movies say, computer programmers don't actually work in 1s and 0s. They work in C++ or something simpler, and they use compilers to generate the computer code. The same specification, importantly, can be compiled in multiple ways. So what I mean by that is that if you take C++ code and you want it to work on Apple OS and also on Windows, you might have to compile it differently for each operating system. And that code should work for both operating systems. This is sort of the magic of compilations. You can take one specification and make it work in multiple contexts. So we want to do the same thing for biology and for, in particular, chemical reaction networks. So biocompiler converts a biochemical specification to a chemical reaction network model using the SBML systems biology markup language format. And I'm going to just highlight biocompiler is not a simulator. It doesn't solve the equations, it just makes them. But I've worked on many simulation packages. Here are a couple of them. And there are many CRN simulators out there. And biocompiler is not the first CRN compiler. There are many inspirations for this work. The DNA strand displacement compilers, many of which came out of Caltech, are direct inspiration. There's rule-based modeling, which is another way of thinking about biochemical systems that's different from CRNs, but related to how biocompiler works internally. And then there's sort of context-specific compilers. These are cases where you can have a specification that compiles to a model, but only in very specific situations. Biocompiler tries to subsume all these by being a framework that can do general purpose CRN compilation. And the way it does this is with an abstraction. Biocompiler takes a specification and abstracts it into three parts. You have the context, which in biocompiler is called a mixture. You have the parts, which in biocompiler are called the components. These are the things you're modeling. And you have the processes or the mechanisms, how things actually work. And all of these are objects, so they can be replaced and interchanged. And so let's go through an example. I'm going to talk about the lac operon in E. coli. This is one of the most well-studied biochemical circuits in history. And so my mixture, my context is E. coli, which in biocompiler, the class name is going to be given here in gray. In some cases, is called a TXTL dilution mixture. Now, if we're working in E. coli, we know some basic things happen. We know there's transcription, we know there's translation, we know there's RNA degradation, there's dilution. So those mechanisms are sort of preloaded in the context. And we also know that there's certain species like RNA polymerase and ribosomes and RNases that take care of how these mechanisms function. Now I'm going to put the lac operon in. This is what we actually want to model. And this is a piece of DNA that has a bunch of binding sites to different transcription factors. And in the presence of these transcription factors, the lac operon can take on different conformations. It can fold. So that the lac R tetramer can bind to these different R sites and actually bind on both sides and induce these different folded conformations. And so in order to model that, you need some new mechanisms. You need to have things like binding to represent the binding of these transcription factors to the operon, as well as polymerases, and conformation changes to represent how, maybe something unbinds and rebinds, or this thing folds. And you also get some more species here. We know that the lac tetramer has, its tetramer species. We know it binds to allolactose. CAMP is a part of the CCRP complex. And next, you have to add in the other enzymes that are important in this system. So we know that the lac operon codes for 3 genes, Z, Y, and A, one of which is lactose permease. So if the lac operon produces that, it should probably be in our model. We know that the lac operon is dependent upon the amount of glucose in the cell as well. It regulates E. coli metabolism based upon glucose to turn on lactose metabolism. So we know that glucose permease should be in the model. And then beta-galactosidase is an enzyme that breaks down lactose, so that's in the model as well. And you get some more species here as well, like the internal and external glucose and lactose. And when you compile all of these components with the mechanisms, you get a chemical reaction network. And so here's sort of a schematic of that network. Each dot is a formal chemical species, and they are connected by mechanisms to other dots, meaning that they somehow interact with other species. For example, if you look at this big set of light blue dots on the far right, those are all the open conformations of the lac operon, but maybe different things can be bound to different places, and all of these conformations can be transcribed to produce an RNA. And what biocompiler does is it takes a fairly simple specification, about 50 lines of code to describe how all these components interact, and produces a much larger model with 173 species and 343 reactions. So that sort of shows you what a biocompiler specification looks like, but more than just being efficient in that sense, biocompiler is also, I think it's well-designed software. It's very modular. So you can generate architectures by reusing components. For example, I might have this transcriptional unit called a DNA assembly that represents the single gene that can be transcribed and translated. And if I combine two of them, I can get inducible repression, where a repressor represses a reporter. If I have A repress B and B repress A, so now I have two things, but they both talk to each other, I can get the bistable toggle switch, a very famous circuit from synthetic biology. If I combine three of them where A represses B, B represses C, and C represses A, I get the repressilator, another famous circuit from synthetic biology. Biocompiler is also expressive. So not only can I recombine components in different ways to generate architectures, but I can try out different implementations and different levels, different mechanisms by which things can occur. For example, I can have this idealized repression, or I can think, well, in the lab, maybe I want to build a repressor that uses a dCas9 enzyme. This is if you've heard of CRISPR-Cas, this is one of those enzymes that couples to a guide RNA and can then repress a reporter. So this is how you might actually build a repression circuit in the lab. Well, now you have 3 parts instead of 2, and it makes the dynamics a little bit more careful. And it turns out you have to have, about the same amount of dCas as you do the guide RNA for things to work. Makes sense. Otherwise, you don't get the repression you expect. I can take my transcription factorial toggle switch I showed you in the previous slide. Here we're repressing transcription, and I can ask, could I build a toggle switch that represses translation, perhaps by degrading RNAs? And the answer is, well, at least in simulation, yes, you could. So here is a theoretical targeted RNase toggle switch where you have 2 RNases that can degrade each other at the RNA level instead of at the transcriptional level. So Biocompiler lets you explore all of these ideas very easily. You can also manage complexity. So what I mean by that is I can test model assumptions and simplifications. For example, when you're building models, it's not always clear how much detail you need. Sometimes you might want a lot of detail. Sometimes you want to use simplified equations. And one way to figure out the right answer is to just try a bunch of things and see which ones work. And Biocompiler makes that quick and easy. So if I look at the repressilator, again, in this model, I'm neglecting all biochemical machinery. There's no ribosomes and no polymerases. It's just sort of a very idealized model of how this circuit works. But if you actually look at electron micrographs of ribosomes bound to transcripts, you can see the ribosomes are the black dots, and there's many ribosomes bound to one transcript. This is called multiple occupancy. And you might ask, does multiple occupancy affect how the repressilator works? Well, I can build a multiple occupancy model where now multiple ribosomes combine to one transcript and multiple polymerases combine to one gene, and I can simulate it, and I still get oscillations. That's pretty cool. That says that the simplified model is a reasonable assumption, even though the biochemistry is actually much more complex. And perhaps most importantly is you can do all these things, but they're very, very efficient and easy to do. So if you look at the size of my models, either in the number of species in blue, the number of reactions, or you can imagine writing out the models as a bunch of ordinary differential equations, how many terms are there in these equations? You see that in green. You can compare that to the number of lines of code you need to generate the model. And the answer is you can generate hundreds of species and hundreds of reactions and many more ODE terms in about a dozen lines of code, which is to say Biocompiler is very efficient. Now, given that I can generate all these models, the question is what models are interesting? Where should I be looking? I can build any model I want easily now. And one way to sort of think about this problem is that theory can guide me and can help me figure out which models matter. So I'm going to talk about some theoretical models now and sort of switching gears. And what I'm going to do is try to show you some biochemical models that are related to machine learning. And the machine learning algorithm I'm going to really focus on is called a Boltzmann machine. And so let's talk about what a Boltzmann machine is and why I'm choosing this as sort of my prototypical machine learning algorithm. So mathematically, a Boltzmann machine is a probabilistic machine learning algorithm, meaning that it uses probability distributions, P of X. And you can see the dynamics of a Boltzmann machine over here. You'll see these black dots are turning on and off. When they're filled in, they're on. When they're empty, they're off. And they're sort of turning on and off randomly or stochastically. If you watch this for a very, very long time, you get some probability distribution of each state, where a state is the set of dots that are on or off. And that's given by 1 over the partition function e to the negative energy function, where the energy function is quadratic. Basically, you can imagine these W's. If W is positive, Xi and Xj don't want to be on together. And if W is negative, Xi and Xj want to be on together. And then you have bias terms that sort of bias each individual node. Now, the reason why Boltzmann machines are interesting is because they can do many things we want a computational system to be capable of. They can represent very complex distributions. And I'll give you an example of that in a second. They can compute conditional distributions, which is inference. And so inference is a very powerful computation. I can ask, given some priors, what do I think? That's a form of inference. That's a very general question. We want computers and maybe cells to be able to do that. How do Boltzmann machines compute inference? Well, they do it by holding some of the nodes constant and letting the other ones stay free. So you see here, the blue nodes are held constant, they're not changing, but the black nodes are still allowed to change. This computes a conditional distribution, P of V, the black nodes, given H, which are the blue nodes. And finally, Boltzmann machines can learn. And so here are some equations that are sort of the stochastic gradient descent learning rule for a Boltzmann machine. I'm not going to talk about these in great depth, but importantly, given data, I can choose my parameters, W and theta, to best match that data via an algorithm. So the third chapter of my thesis talks about chemical Boltzmann machines. And what we do is and this is with a bunch of co-authors, Andres Ortiz-Munoz, Nick Jones, Tom Oldridge, Erik, Winfree, and Manoj Gopalkrishnan. We show 3 different chemical reaction network implementations of Boltzmann machines. I'm going to go over these very, very quickly. The first is the direct chemical Boltzmann machine, which exactly models a Boltzmann machine. The second is the edge species chemical Boltzmann machine, which could have different dynamics from a Boltzmann machine, but has exactly the same probability distribution or steady state distribution. And the third is the Taylor chemical Boltzmann machine, which approximates Boltzmann machine dynamics and approximates the steady state. So for example, you can think about Boltzmann machines in terms of what computation they do. This is back to that data set I mentioned, MNIST, where we have digits 0 through 9, and we put in a digit and we ask the Boltzmann machine, what digit are we looking at? So if you give it a 2, it thinks most 2s are 2s, although occasionally it confuses 2s and 0s. If you give it a 5, it gets a little more confused. It confuses a lot of 5s with 0s, but it gets some 5s right. And this is what a Boltzmann machine does, or equivalently the direct CBM or the edge species CBM. These are all equivalent and would have the same classification. The Taylor CBM isn't quite as good, but you can still see that there's sort of a dark line around the middle saying it's doing, fairly well considering that it's an approximation. You can also use Boltzmann machines to generate digits. So here again, I'm showing the digits generated by a Boltzmann machine. Instead of giving it an image and saying, what is this image? I say, show me a 0. And the Boltzmann machine does, it shows me a 0. Or I say, show me a 7, and it shows me a 7. These are a little bit blurry because we trained it on 10 by 10 images, so that they've been downsampled quite a bit. But even the Taylor CBM still sort of works. If you squint really close and look at only the darkest pixels, which I'm showing you down here, the Taylor CBM also shows 0s, 3s, 4s that look like 9s. It does a pretty good job. What this is to say is that chemical reaction networks can do Boltzmann machine-like things. They can work as probabilistic models that can compute inference in many different ways. And I hope that maybe you are beginning to think, okay, if a chemical reaction network can be a Boltzmann machine in not just 1, but 3, and in fact, more than 3 ways, maybe this is a good way to think about what cells and biochemical systems, which can be represented as chemical reaction networks, might be doing. And in fact, chapter 4 of my thesis really tries to dive into this. So it takes the edge species Boltzmann machine construction and tries to expand it. And instead of asking what CRNs implement Boltzmann machines, it tries to say, how can we interpret a whole class of detailed balanced chemical reaction networks in the same way we interpret Boltzmann machines? So it sort of goes the other direction. And let's explain what detailed balanced chemical reaction networks are first. So the intuition is that detailed balanced CRNs are undriven, meaning that no energy sources such as fuel molecules power the system. So there's 2 examples of this. One is molecular binding. A plus B forms some complex C. If A and B just bind together and unbind and there's no chemistry changing, this is a detailed balanced network. Conformation changes, like maybe a protein folding or unfolding or moving, could also be seen in some cases as examples of detailed balance systems, where say we have three conformations, S1, S2, and S3, and we can transition between them. Now, non-detailed balance systems are much more common than detailed balance systems because there's a lot of fuel going on, especially in biology. So any CRN that is driven by implicit or explicit fuel molecules is not detailed balanced. And so two examples, one is phosphorylation. So if ATP phosphorylates some protein X to produce a phosphorylated version of that protein and becomes ADP, well, ATP is acting as a fuel molecule here, and therefore this is not detailed balanced. Similarly, transcription, if a gene produces a transcript, well, there's a whole bunch of implicit fuel molecules. They're all of the NTPs that are polymerized together to form the transcript. So this is also not detailed balanced. But detailed balance networks are still very common. Again, molecular binding, conformation changes, these things occur all over the place. Now, mathematically, we can define this as well. Technically, a detailed balance CRN has this particular mass action propensity function, while noting we're talking about stochastic CRNs here, so and probability distributions. All reactions are reversible, so if I have some inputs that go to an output, that means the outputs can also go to the inputs. And then you have this microscopic reversibility condition, which is a condition on the relative rates between a forward and backwards reaction is given by the change of energy. So every species in the network has some energy, GI for species SI, and reactions are constrained and their rates are constrained by the change of energy in the reactions. Now this seems like maybe these are strong constraints. In some ways they are, but they're also very physical. These constraints really come out of the detailed physics of how we expect chemistry to behave if you look with enough resolution. Now, we know some things about detailed balanced chemical reaction networks. One famous theorem is called the product-Poisson theorem. And it says that all detailed balance have an equilibrium steady state distribution. So this is the distribution. If you let the network, the CRN, run for a very long time, you get this distribution, pi of S. And let's unpack this. So Z is called the partition function. And it's given by this equation. This gamma of S naught is called the reachability class, and this is very important, so I'm going to explain what it is. It is a function that describes the states S reachable by some sequence of reactions. So I start at state S naught, and I take some sequence of reactions, any sequence, it doesn't matter, in my CRN, and I can get to some other states. And if I enumerate all possible states I can possibly get to, that's the reachability class. So for example, I mentioned this earlier, we have this reaction A goes to 2B. The reachability class of A is just 0 through 10. And the reachability class of B is where these lines come in. It's 0 through 20, but only the even integers. I can never have an odd value of B. So reachability classes can have a lot of structure. And finally, there's an energy function, G of S, which has this linear term and then this log of a factorial. And a product-Poisson distribution looks like a blob in space. So if your reachability class is the integer lattice is what you see. And this is actually done from a CRN. So here you can see a trajectory. These are sort of species counts changing over time. And when you look at two of them, you get this blob. Now I want to highlight sort of some intuition for why we're comparing detailed balanced CRNs to Boltzmann machines. And some of this just comes by looking at the equations. If you look at the equilibrium steady state distribution of a detailed balanced CRN, you get this Boltzmann-Gibbs form. If you look at the equilibrium distribution of a Boltzmann machine, you get something that's very similar. The partition functions are different, the energy functions are different, but the form is very, very similar. And that sort of is a hint that maybe these two systems are related more fundamentally than just, more fundamentally than just CRNs being able to implement Boltzmann machines. And I'm going to explain how that is the case. So one of the things I mentioned that Boltzmann machines can do that we want to be able to do as a model of computation is represent complex distributions. And in my thesis, and a paper that's not yet published with Tom Ultridge, Manoj Gopalkrishnan, and Erik Winfree that we're preparing, we prove that restricting the reachability class is necessary to represent complex distributions with detailed balanced CRNs. So the intuition here is that that product Poisson I showed you before is an uncorrelated blob in space. And we can measure the distance between a detailed balanced CRN's distribution and a product Poisson distribution using the relative entropy. And the idea is that if you have large relative entropy from product Poisson, your distribution is complex. It's somehow much more than a blob in space. To make this distance large, I'm going to prove that many states must be unreachable, or really a high density of states must be unreachable. So here's the theorem. Rho of S is a product Poisson distribution on the integer lattice, and pi of S is the equilibrium distribution of any detailed balanced CRN, which is a subset of the integer lattice. That's its reachability class. Then the relative entropy is given by this formula and is equal to the log of the ratio of these two partition functions, where z rho is the partition function for the Poisson distribution and is summed over the entire integer lattice. And z pi is the partition function for the detailed balanced CRN, and it's summed over just the reachability class. Notice that things that we're summing, e to the negative g of s, are the same in both cases. So the only way I can have this ratio be large is for z rho to be much larger than z pi. And that means that these sets, the integer lattice, well, that's set. I can't change what that is. That's sort of defined as it is. But I can make gamma smaller. If gamma doesn't contain very much of the integer lattice, it is likely that z rho is going to be much larger than z pi. And in particular, you might want to choose the states that aren't reachable. They have to have, be highly weighted states to make this particularly true. So again, the intuition here is that by restricting my reachability class, I can make complex distributions. What does that look like? Oh, this is an important comment for those of you who think about machine learning and latent variables and hidden species. We have a follow-up theorem that says that marginalizing over hidden species only matters if the reachable states of the visible and hidden species are entangled. And I'm using the word entangled loosely here, but we define what that means in my thesis. So let's give some examples of this. Here's a product Poisson distribution from a CRN. So I have nothing goes to S1 and nothing goes to S2. You can see a trajectory up here, and the entire integer lattice is reachable. You're seeing that with this white line. It goes off to infinity. Everything, any state can be reached, but my distribution is a blob in space. I'm most likely, around here. However, with the fairly simple constraints, now I have a slightly different CRN. S1 goes to S2 goes to S3. I get a very different looking distribution. Now here my reachable states are constrained. Instead of having the entire integer lattice, I have this constraint S1 plus S2 plus S3 is equal to some constant. And just by having that simple constraint, I'm inducing correlations or anti-correlations between these species. Now, if I use a very complicated CRN, this construction is from this paper with Erik, Winfree, and co-authors. We can make actually any arbitrary finite distribution. And if I write down the constraints algebraically, how the reachability class is constrained, you get some very complex equations. What this is to say is that the reachability class of this CRN is very, very highly constrained, compared even to this one, and certainly compared to the product Poisson. It's so constrained, in fact, that we can program it to represent any distribution that's finite, like this little emoji face. So I hope you're convinced that detailed balanced CRNs can represent complex distributions. The next thing we like about Boltzmann machines is that they can do probabilistic inference. What I mean by that is they can compute conditional distributions. So to give some intuition, Boltzmann machines compute inference by holding species constant. So H is held constant here. In a CRN, we can't hold species constant. This can actually have some problems, can change the reachability class, and is generally mathematically intractable. Instead, what we do is we're going to develop a way to hold the mean values of species constant, and we're going to prove that this is a type of conditioning, a type of inference that's applicable to any detailed balanced CRN. So here's the theorem. I start with my energies GI for each species, and I'm going to modulate them. I'm going to add some delta to some of them. These are the clamp species, the ones I want to hold their means constant to some value. I add delta. If I add a positive delta, I'm going to push the counts down. If I add a negative delta, I'm going to make the counts go up. And this gives me some new clamped energy function, GI clamped. This new clamped energy function can be put back into the same CRN I had before to get a new detailed balance CRN with the new energy function that has an equilibrium distribution, a new partition function, a new general energy function. Again, all that's changing here is this GI is being replaced with GI clamped. The reachability classes, importantly, are the same. And you can prove, I'm not going to prove it for you here, but using the conditional limit theorem, which is a sort of theorem from information theory, that pi c, the clamped equilibrium distribution, is equal to the unclamped distribution pi of s, given that the mean value of the clamped species is equal to c bar. Now, c bar here is a constant that's either determined by delta. So given if I choose delta, I will get some c bar, or I can choose c bar and then find the right delta. Those are both possible. And the point here is that clamping, changing these energies, is equivalent to inference, because I just showed that pi of c is a conditional distribution, and conditioning is inference. That's what we're trying to get at. So now, okay, maybe it's conditioning and doing inference. Maybe it can't do complicated inference. Well, it can. You can do some pretty cool things with this. So here's some examples. S1 goes to S2 goes to S3. I showed you the CRM before. I'm going to just clamp S3. So you can see down here in these histograms the marginals. So here I've made S3 unfavorable. So S3 goes towards 0, and S1 and S2 are very anti-correlated. As S3 becomes more favorable, as its energy decreases, the correlation or anti-correlation between S1 and S2 decreases as well. So when S3 is very favorable, S1 and S2 are almost not anti-correlated anymore. This is almost a blob in space, which is to say that conditioning upon a third variable, S3, can induce changes in the correlations or this, in information about S1 and S2. This is a pretty sophisticated computation. And it can get even more sophisticated. So here I'm taking that emoji CRN I showed you before, and I have a more complicated version of it. And by clamping a single variable C alpha, I can go from a frowny to an open mouth to a smiley, just by making C alpha more or less favorable. And this is, really changing my distribution in some very nonlinear ways. And I'm inducing these changes by clamping a single species. So the power of this type of conditioning, this type of inference in detailed balanced CRNs is pretty high. You can do a lot of computation. Now, I also want to bring this back to biology. One of my favorite examples of energy clamping is applied to polymer conformations. Now, polymer conformations are what I call a polymer, like a piece of DNA or a protein that maybe can form some, can connect to itself, bind to form something like a hairpin. Maybe it binds in multiple places, form some secondary structure. I'm calling that a polymer conformation. And in this case, I can write this as a CRN. S open goes to S looped. So that's open, and this is the looped version. And if I change the sequences that are involved in binding, maybe this is like, a piece of RNA and you're forming a hairpin, that changes my binding energy, which is to say I'm changing an energy by changing a sequence. The evolution of sequences and how they change binding energy can then be interpreted as a type of energy clamping and inference. Now, I think this is something that would have to be elaborated on in the future, but certainly it is suggesting that there's probabilistic and inferential ways to think about evolution. Now, the thing I'm actually even more excited about is another way of implementing energy clamping, and you can do that with an external chemical potential. So what I mean by those fancy words is that if I have the same loop CRN, but in this case, some molecule P, which we call a potential species, has to bind to form the loop, by putting more P in solution, I can make the looped complex more favorable, and by having less P, I can make the loop complex less favorable. And we have an equivalence theorem showing that these two cases are identical in many situations, namely that I can implement energy clamping with a particular set of potential species coupled to the CRN in the right way. And this is very cool because this allows for an external chemical potential to dynamically do inferential computations. I can be changing the concentrations of P and changing, my energies, dynamically computing probabilistic inference. And perhaps most excitingly, which I'm not going to talk to you about, is that you can tie all of this together and have a chemical reaction network which tunes itself. And this is an autonomous learning system. And just to sort of say, you start with a network that doesn't know anything, and in the end, it learns some distribution modeled after a complex environment. And because this entire system is one CRN, you can do things like prove some thermodynamics costs related to learning and understand the actual physics of machine learning, at least in this model. So I refer you to my thesis or the future paper to learn more about this. What I hope you've taken away from all of this discussion is that detailed balanced CRNs can be thought of as generalized Boltzmann machines. They're a probabilistic machine learning algorithm versus a probabilistic chemical reaction network model. But it's ubiquitous in biology. So you see detailed balanced CRNs all over biology. They can both represent complex distributions. They can both compute conditional distributions, which is to say probabilistic inference. And they can both learn. In the case of a Boltzmann machine, they learn using stochastic gradient descent. And in the case of detailed balanced chemical reaction networks, they can be coupled to auxiliary species to learn as a CRN, or also you can do it in silico with stochastic gradient descent. Now, given all of this theory, it seems kind of abstract. Does it actually help us understand biological systems? So I'm going to very quickly touch on a couple of theoretical things I've done during my PhD. The first thing I'm going to talk about is this cell extract protein expression experiments. And briefly, cell extract, what is it? I take E. coli and I lyse it. I break all the cell membranes. You then pull out the native DNA and the plasma membrane. So you're left with a bottle or typically a small amount of green goo, literally. That is the guts of E. coli. It has all of the proteins and much of the small molecules, but not any of the genome or membrane. So it's not alive, but it has a lot of the complexity that's in E. coli. I think it's a very interesting place to work because you get some of the hard parts of biology, like a lot of unknown variables and quantities, but also some of the hard parts of biology are not there, like my system isn't going to evolve away on me. And in a cell extract protein expression experiment, you combine energy buffer, which is mostly 3-PGA, which is a fuel molecule related to pyruvate, with extracts and maybe some sort of DNA circuit that can then run in this in vitro setting. So you get like a fluorescence output, for example, can be measured in a plate reader. You can put in all kinds of things here. Now, I chose I wound up looking at the metabolism of E. coli cell extracts because there was sort of an opportunity then and lots of questions. So there have been many models of how cell extract works in the transcription translation area. So, how does a gene go to a transcript, go to a protein? There have also been some heuristic models of energy utilization, namely how are these processes powered by fuels like the energy buffer? And there's definitely specific metabolic models of parts of metabolism, but they haven't been integrated very well with gene expression. So in fact, when you think about these fuels, my ATP goes to ADP and is then regenerated. So there's sort of a reverse pathway here where my food, like 3-PGA, is used to regenerate some other fuels. And this has not been systematically modeled in cell extracts, but it's something that really matters. If we could understand cell extract metabolism better, we could understand how circuits perform in cell extract and use it as a better prototyping platform. And B, you could start using cell extracts and engineering cell extracts to make as biochemical production machines. But to do either of those things well, we have to understand and be able to quantitatively model cell extract metabolism. So to do that, I did some metabolomics. So what is metabolomics? It's a type of untargeted high throughput mass spectrography, which measures the abundance of many small molecules at once. So basically, I'm trying to get lots of measurements. The goal here was to get enough measurements to maybe do some machine learning. And so to give you an example, 87 of the 328 measured metabolites vary significantly in time. And you can see sort of them here. Some of them start dark and go light, some start light and go dark, meaning that some things are being consumed and other things are being produced. And if you do some statistics, what you see is that you can combine these metabolites at sort of a pathway level, and you see that 47 out of 55 metabolic pathways also vary significantly in time. So what does that mean? That means that most of E. coli's metabolism is active in cell extracts. And these other columns, you can see here is the time course. These other columns are different conditions. Most conditions we tried didn't change metabolism very much. So, how complex is E. coli's metabolism? Can we build a model of this? Well, if you look in the literature, E. coli has over 1,000 metabolites, over 1,000 genes code for metabolic enzymes, and there's over 2,000 reactions and a whole bunch of unknown regulation. We don't really know how all of these different parts talk to each other. And this is one of the best studied organisms in history, and we don't really know how it's regulated. That's, to me, a little crazy. How big is my metabolomics data set? I have 12 conditions times 4 time points times 327 metabolite signatures. That's about 15,000 data points. Now, this is about as big as you can easily get. I had a company, Metabolon, do the metabolomics for me. They have one of the best libraries of standards. They use the best equipment available. You can't do much better than this. You could do more time points and more conditions maybe, but they're basically getting almost as much as we know how to get right now. Let's compare that to MNIST. MNIST is the digits 0 through 9. Here's some examples. The data set of MNIST is 60,000 labeled images, and they're each 28 by 28 pixels, which amounts to about 47 million data points. So and this is sort of what it takes. MNIST is probably bigger than we need to understand the digits 0 through 9, but it's about the right order of magnitude. We have 3,000-fold less data points in this metabolomics data set than we do in MNIST, and we're trying to understand something that's probably much more complicated than the data 0 through 9, which is to say that this data set's not big enough for machine learning. What I could do with it, which I won't talk about in detail, is use it to figure out a heuristic phenomenological model of how metabolism works that I think is better than what we currently have. But really what it taught me is that we need to do something new in order to bring big enough data sets to biology in order to apply the theory and computation I've been working on. So on that note, I want to conclude with sort of wrapping everything up in a totally different way. I hope I've convinced you that probabilistic inference is a model of cellular computation. You can imagine a cell in some environment and see some noisy information, and it's going to make inferences about its environment, try to make sense of it in a way that might be like probabilistic inference. There's lots of ways this could happen. In particular, detailed balanced CRNs are probabilistic inference machines. They are particularly well suited to probabilistic inference, and polymer conformations of the space of detailed balanced CRNs are one example of a system that exists in biology and can do probabilistic inference. Now, I showed you that, in this very well-studied model of the lac operon, it's actually polymer conformations that are controlling this small piece of metabolism. So you see here that there's these 3 conformations. They look a lot like the picture I drew here. And that wasn't on purpose. That's just how the systems work, which is to say that maybe we can think about metabolism as some sort of, or at least governed at some level by probabilistic inference. Now, what does that mean with respect to the metabolomics data I gathered? Well, we saw that all of metabolism roughly is active in extracts, but maybe that's sort of a population level effect. So just by taking this viewpoint that cells could be doing probabilistic inference, one hypothesis I can come up with is that each individual cell has its own unique polymer conformation that has some subset of metabolism active. In some cells, they're open and lac metabolism is on. In other cells, it's closed and lactose metabolism is off. When I combine, average all of these cells together, it's going to look like everything is on because I'm averaging over a large millions, billions of cells. But at the individual cell level, maybe that's not the case, which is to say that just by thinking about cellular regulation as a machine learning algorithm, I can start making hypotheses about about data that we actually gather. And in particular, you can start thinking about how does this help us improve metabolic capabilities of extracts? Well, if I think about back to this inference, a cell is inferring things about its environment and conditioning its internal state to best match its environment. If I change the external state of the cells as I grow them, maybe I'm going to get different metabolic capabilities for my extracts. And somewhat surprisingly, not a lot of people have spent time changing the media, how they grow E. coli to make cell extracts in order to change their fundamental metabolism of the extracts. This seems to me now a natural thing to do, but before I started writing my thesis, I'd never considered it. So the punchline is taking the perspective that cells do probabilistic inference as a computation really can actually add to how we interpret, understand, and design new biological experiments and systems. So that's the end of my talk. I wanted to mention a couple of acknowledgements. This PhD would not have been possible without a great many people, particularly the guidance of my advisors, Erik, Winfree, and Murray. And Erik's right, I should put Manoj here, Gopalkrishnan. He's been a great source of mathematical wisdom through this process. Obviously, the support of my wife, Abha, parents, Will and Janet, and sister Sarah. There are many collaborators on all of these projects. I've listed them out here. I don't think you can work alone in science, do cool things, and I've worked with a great many very smart, talented people who've helped me on all of this. And the support of all my peers and friends in the Murray Lab and DNA Supergroup and Caltech at large. Thank you. Many of you are here. I really appreciate that. And also all my past teachers and mentors who helped me get here. I also see some of you in this room, which is really makes me feel good, and I really appreciate you coming. So thank you, everyone, and I hope you learned something today, and it's now time for some questions. I'm also going to stop recording at this point.
Okay. Thank you, William. That was truly fantastic. So I do hope that we'll have plenty of